We're now using brain organoids and culture them in a high throughput manner, which will allow us to test the effect of different drugs and how they are suitable to stop brain cancer invasion, but also investigate what are the genes that are activated once the cells become invasive.
So clearly the only way we can address this issue of brain cancer is working as a team, and this project is all about teamwork. We are integrating different expertise from different institutes in Adelaide, including the South Australian Health and Medical Research Institute, SAHMRI, the Centre for Cancer Biology at UniSA, Flinders University, SA Health. And what's really unique about this project is the collaboration between different teams with completely different expertise. Guillermo Gomez has tremendous expertise in cellular mechanics and microscopy. My group has specific expertise in stem cell biology and neurological models using stem cells. And we also be working with obviously clinicians and chemist and mechanical engineer.
To create a mini brains that can be grown in a high throughput platform, so it's absolutely needed the capacity to grow so many mini brains in the dish, and that is only possible by combining material science, novel approaches, and this is collaboration with Dario Arrua in the Future Industries at University of South Australia. But also another challenge is if we want to do some drug screening for personalized treatment, we need to be able to analyze all that data in one single go. So for that we collaborate with Chunhua Shen. He is a top scientist in machine learning and computational analysis of images. So he combined his expertise into this project, and actually that will speed up all our capacity to generate rapid results.
So the advantage of having a research team coming from many different disciplines is that we can translate a project from the bench in the laboratory here at CCP to the bedside of a patient at the Royal Adelaide Hospital, where there's actually need to do something for brain cancer in this particular case. And it's only possible really with the expertise of each of the scientists and the clinicians working on the project.
We're very well aware of the urgency of finding treatment for cancer patients. Once they're diagnosed with the tumor and the surgeon removes the tumor, they usually have around 1 or 2 years of survival on average. So we want to really create a platform that works with that timeline. We want to be able to get results after getting the surgery from the patients within a few months, and be able to guide the doctors to find the right treatment for that. The limitation that clinician and patient have within that short time frame is they can only try maybe 1 or 2 chemical therapy. And sometimes they work, sometimes they don't work. And each patient is unique in many ways. So with that platform, we will be able to preclinically test maybe tens, maybe hundreds eventually of treatment in the cells of the patients in the lab before we give them to the patients in the clinics, saving times and hopefully have better success and better outcomes. Of course, we want some aggressive treatment that affect the cancer cells, but we don't want to damage too much the neurons that are healthy in the surroundings. So we'll be able to assess both effect of the drugs with that model.
We're very pleased to get this grant from Cure Brain Cancer Foundation. It's going to be very important for us to develop a resource for patients so that we can take brain tumour material from patients and put it in the test tube and see how it behaves with normal brain tissue.